Welcome back to the deep dive. Now, if I were to ask you, what is a carbohydrate? You'd probably jump straight to energy, right?
Uh-huh.
Calories, maybe just simple sugar.
hmm
But the stacks of biochemistry sources we dug into this week, well, they paint a totally different picture. These molecules, they're way more than just fuel. Think molecular architects, identity badges.
Absolutely. Even natural shock absorbers inside your own body.
Exactly. So our mission today really is to get past carbs as just, a diet thing
Right.
and decode this incredibly complex chemical language of saccharides.
That's the goal. And if we start, sort of fundamentally, the classic chemistry definition is pretty neat. Carbohydrates are polyhydroxylated compounds.
Meaning lots of those OH groups.
Lots of OH groups, yeah. And they're either aldehydes, called aldoses, or they're ketones, the ketoses. That's the basic chemical split.
Aldoses versus ketoses.
Yeah.
And what jumps out immediately looking at the sources is just the sheer variety. So many names.
Oh, absolutely. But there's a logic to it, mostly based on the carbon count. The simplest are trioses, 3 carbons. Then you get pentoses, 5 carbons, think ribose, super important for RNA.
Okay.
And then the big ones, the hexacess is 6 carbons. That's where you find glucose, galactose, fructose,
Yeah.
the famous ones.
Okay, let's unpack this. Because the first layer of complexity, even with these basic building blocks, is how structure equals identity. Tiny differences in 3D space matter hugely.
That's where it gets really interesting. It's all about stereoisomerism, which comes down to chirality.
Chirality.
Like left hand, right hand. Exactly like your hands. They're mirror images, but you can't perfectly overlay them. So molecules can exist in D and L forms, and this distinction is massive in biology.
How so?
Well, think amino acids. The building blocks of proteins, they're almost all L isomers in us. But sugars, virtually all the sugars we use and find in our bodies are in the D configuration.
So our enzymes are basically built to recognize only the D handshake.
Precisely. They're shaped for it. That DVSL difference is fundamental.
That alone is pretty mind-bending. But
Yeah.
even within those D sugars, the sources talk about more structural differences, right? Enantiomers first.
Yeah, enantiomers are the perfect mirror images, like D-glyceraldehyde and L-glyceraldehyde, non-superimposable, just like your hands.
Okay.
But then you get the epimers. Epimers are maybe the real tricksters here. Same chemical formula, same atoms, but they differ in the 3D arrangement around just one specific asymmetric carbon atom. They're diastereoisomers, technically.
So if we're talking chemistry, what does flipping just one carbons group actually mean inside the body?
What's fascinating here is how such a tiny tweak creates a totally different compound biologically.
Like glucose versus mannose?
Perfect example. D-glucose and D-mannose, they're identical except for the direction of the OH group at carbon number 2, just C2.
Wow.
Or glucose and galactose, they only differ at carbon 4, C4. Flip that one hydroxyl group and bam, you have a molecule with a completely different biological identity.
Different enzymes needed, different pathways?
Exactly. It dictates how it's stored, how it's used, how it's recognized by other molecules. It's chemical precision down to one atom's position.
Okay, that's structure. But then there's the shape they take in solution. We always draw glucose as a straight line, but the sources say that's misleading.
Very misleading. In water, which is, basically everywhere in our bodies, that straight chain form is unstable. Sugars like glucose fold up on themselves.
Into rings.
Into rings, yeah. It's much more stable thermodynamically. Aldohexases, like glucose, usually form these 6-membered rings called Pyranose rings. Fructose often forms a 5-membered furanose ring.
And closing that ring creates another complication, doesn't it? Something called the anomeric carbon.
It does. The ring closure creates a new center of asymmetry, usually at carbon 1 in aldoses. And this gives rise to 2 possible forms, the alpha and beta anomers.
Alpha and beta glucose, I remember those.
Right. And for D-glucose, the beta form, the beta pyranose, is actually the most stable and the most common.
Why is that?
Because that crucial hydroxyl group on C1, the anomeric carbon, ends up in what's called the equatorial position. It sort of sticks out to the side, away from the bulk of the ring, less crowded.
So in our blood, or anywhere watery, glucose isn't just one thing. It's constantly shifting between that tiny bit of open chain, the alpha form, and mostly the stable beta form.
That constant shifting back and forth, that dynamic equilibrium, is called mutar-rotation. You've got maybe 62% beta, about 38% alpha.
And less than 1% open chain.
Less than 1%. But, and this is crucial, that tiny fraction of open chain is still incredibly important.
The sources really hammered this point, especially for clinical stuff. Why does that tiny bit matter so much?
Because only the open chain form still has that reactive aldehyde group exposed, and aldehydes get oxidized easily.
Oh, making glucose or reducing sugar.
Exactly. Its ability to reduce other substances like copper ions was the entire basis for those early diagnostic tests for diabetes.
Like Feelings or Benedict's.
Benedict's reaction, yeah. You could literally see the color change as the glucose in urine reduced the blue cupric ions to reddish cuprous oxide. That simple redox chemistry gave doctors the first real way to measure sugar levels.
It's amazing how stability and that tiny bit of instability both play these vital roles. Okay, so the single sugars. But the real engineering starts when they link up, right? Forming chains.
Right, the DS and polysaccharides. Monosaccharides link together through a dehydration reaction. Water gets removed, forming what's called a glycosidic bond.
And this bond connects the anomeric carbon of one sugar to an OH group on another.
Usually, yes. And how they link which carbons are involved and whether the bond is alpha or beta that literally writes a code. It determines if the molecule is sweet fuel or tough structure or something else entirely.
You see that code clearly in the simple disaccharides, like maltose, that's two glucoses linked.
Okay. Maltose uses an alpha-1,4 bond. The anomeric C1 of one glucose links to the C4 hydroxyl of the next in the alpha configuration.
Okay, and lactose, milk sugar.
That's galactose linked to glucose, but this time with a beta-1,4 bond. Different sugar, different bond orientation.
And sucrose, table sugar.
Sucrose is different again. It's glucose linked to fructose using an alpha bond from glucose's C1 to fructose's C2, an alpha-1,1,2 linkage.
And there's something special about that sucrose linkage, chemically.
There is. Because both anomeric carbons C1 of glucose and C2 of fructose are tied up in that bond, neither ring can easily open up to expose an aldehyde or ketone group.
Meaning
Sucrose is a non-reducing sugar, chemically distinct from maltose or lactose in that way.
Got it. So building blocks, check. Linkages, check. Now, the big polymers. The source is really focused on the classic comparison. Starch versus cellulose. Both just glucose chains, right?
Yeah.
But one's food, one's fiber.
This is probably the biggest aha moment in basic carbohydrate chemistry. It's all glucose, yes. But the difference, the profound functional difference, comes down entirely to the orientation of that single glycosidic bond.
Starch first. That's the energy storage in plants. That's its linkage.
Starch uses alpha-1 4 linkages primarily. This forms amylose, which coils up into a helix great for packing energy away. Then you have amylopectin, which adds alpha-1 6 branches for quicker access.
And glycogen are storage form in muscles and liver.
Same chemistry. Alpha-1 4 and alpha-1 6 linkages, just even more branched than amylopectin, allows for really rapid glucose release when we need energy fast. The alpha bond makes it accessible fuel.
Okay, so alpha means fuel. Now, cellulose, the single most abundant organic molecule on Earth. Plant structure, wood, cotton.
Also just long chains of glucose, but here every single link is a beta-1 4 glycosidic bond. No alpha bonds at all.
Beta-1,4. If alpha is like a regular handshake, beta is...
Maybe like trying to shake hands with your knuckles facing the wrong way. It creates a very different shape. Instead of coiling, the beta-1 links let cellulose chains lie flat and straight. They stack up, form hydrogen bonds between chains, and create these incredibly strong, rigid fibers.
And that single difference, alpha versus beta, defines our diet.
Absolutely. Our digestive enzymes, like amylase, are built to break alpha-1 4 bonds. Effortlessly cuts up starch and glycogen into glucose fuel.
But the beta-1,4 in Vond.
We don't have the enzyme, cellulase, needed to break that specific beta-1 4 linkage. So for us, cellulose passes straight through. It's indigestible fiber, roughage, starch is energy, cellulose is structure.
All down to one bond angle.
Yeah.
That's the kind of subtle power these sources kept highlighting.
You see that beta-1 structure elsewhere too, like in chitin.
Chitin, that's in insect shells. Fungi.
Exactly. It's the structural polysaccharide in arthropod exoskeletons and fungal cell walls. It's not glucose, but a modified glucose called N-acetylglucosamine, linked by those same strong beta-1 bonds. Same principle. Structure, not fuel.
Okay, so beyond fuel and basic structure, carbs get even more diverse when they're modified, right?
Yeah.
Or attached to other things. They become informational.
That's the next big leap. We move from bulk material to specific signals and complex structures. This involves functional derivatives.
Like sugars with amino groups added. Or charges.
Precisely. Amino sugars like that N-acetylglucosamine in chitin. Or acidic sugars carrying negative charges, like glucuronic acid or neuraminic acid, which you might know as sialic acid. These modifications change everything.
And these modified charged sugars often build up into complex structures themselves.
Yes, they form these long repeating chains called glycosaminoglycans, or GAGs, often highly negatively charged. Key players in the extracellular matrix, the stuff between our cells.
The sources gave a great example. Hyaluronic acid. What's special about it?
Hyaluronic acid is enormous, a huge GAG, and its structure, with all those negative charges and hydroxyl groups, makes it incredibly good at attracting and holding onto water.
Like a biological sponge?
A super sponge. It can hold like 10,000 times its own weight in water. This creates lubrication and shock absorption in connective tissues, in your joints, the fluid in your eyes.
That gel-like stuff in the eyeball, the vitreous humor.
That's hyaluronic acid doing its job. It's biological engineering for resilience.
Wow. And there are other gags too, like heparin.
Right, heparin is another GAG, but heavily sulfated, lots of extra negative charges. This makes it a potent anticoagulant. It binds to proteins involved in clotting and shuts them down. Hugely important clinically, but also occurs naturally.
So gags handle complex structural and hydration roles. But the ultimate information role seems to be when carbs are attached to other things, like proteins or lipids.
Exactly, glycoproteins and glycolipids. These molecules stud the outer surface of our cells. They are, essentially, the cell's communication antenna and identity markers, crucial for cells recognizing each other, interacting, sticking together.
Here's where it gets really interesting, because the classic example of this sugar code defining identity has to be the ABO blood groups.
Absolutely perfect example. Your blood type A, B, AB, or O is determined purely by the specific short chain of sugars, an oligosaccharide attached to proteins and lipids on the surface of your red blood cells.
And the difference between A, B, and O is tiny.
Incredibly tiny. It comes down to the very last sugar added to a core structure. Type A has an N-acetylgalactosamine on the end. Type B has a galactose on the end. Type O just doesn't have that final sugar. It's missing.
And that tiny sugar difference dictates who you can safely receive blood from.
Precisely. Your immune system recognizes those sugar structures as self or non-self. That little sugar chain is a critical identity badge.
Wow. And these sugar badges, they're read by specific proteins.
Yes, by proteins called lectins. Lectins are carbohydrate-binding proteins. They specifically recognize and bind to particular sugar structures. This is fundamental to cell recognition.
And pathogens use this too, right? Like the flu virus.
Exactly. The influenza virus has a lectin on its surface that specifically latches onto sialic acid residues, those charged sugars we mentioned, found on the surface of our respiratory cells. That binding is the first step to infection. The virus reads the cell's sugar code to get in.
Okay, let's look back one last time to glucose itself in the bloodstream. We talked about the open chain reacting. What happens if glucose levels are high for a long time, like in untreated diabetes?
Well, that reactive aldehyde group on the open chain form doesn't just react in diagnostic tests. If glucose levels are chronically high, it starts to react non-enzymatically with proteins all over the body. It just sticks to them.
A process called glycation.
Glycation, yes.
Wow.
And one protein it sticks to readily is hemoglobin, the protein inside red blood cells.
Which gives us that really important diagnostic marker, HbA1c, glycosylated hemoglobin.
That's the one. The percentage of your hemoglobin that has glucose stuck to it, the HbA1c level, gives a measure of your average blood sugar control over the lifespan of that red blood cell, typically weeks to months.
So the sugar literally marks the protein, creating a historical record.
Exactly. It's a fantastic tool for managing diabetes because it reflects long-term control, not just a snapshot like a finger prick test. The carbohydrate chemistry provides the record.
So looking back at this whole deep dive, carbohydrates are just way more complex than sugar. Their function hinges on tiny chemical details. D versus L forms, those single carbon epimer differences, and crucially, that alpha versus beta bond angle separating fuel from fiber.
And beyond energy and basic structure, they become these incredible informational molecules, the charged GAGs acting as shock absorbers.
And the glycoproteins and glycolipids acting as cell identity tags, like the blood types, governing how cells talk to each other.
It's a whole chemical language built from sugars.
Okay, final thought then. The sources hinted at something really profound about glucose itself. Why glucose became the universal blood sugar.
This raises an important question. It wasn't just about energy content. The suggestion is that glucose was evolutionarily selected partly because of its chemical stability. Compared to other similar hexoses, it's relatively less prone to that non-enzymatic glycation we just talked about.
So it's less likely to randomly stick to proteins and cause damage.
That seems to be part of the evolutionary logic. It's stable enough to transport and use, but not so reactive that it constantly gums up the works.
Which makes you wonder, what tiny structural flaw exists in, say, mannose or galactose that made them just a bit too reactive, too prone to sticking to things, preventing them from becoming the main currency?
Was it the precise arrangement of those hydroxyl groups in the ring form, making the open chain form slightly more accessible, or the aldehyde group slightly more reactive?
It suggests chemical stability, maybe even more than energy, was the deciding factor in life choosing glucose. Something to chew on.
Definitely food for thought. Thank you for joining us for the deep dive.
We'll see you next time.